If we zoom in on the rigid cell wall of a plant stem and compare it to a human liver, we find a biological paradox. Both of these structures are homopolysaccharides. They are built from the exact same chemical monomer, D-glucose. Yet the human body reacts to these identical building blocks in completely opposite ways. The liver tissue stores a polymer that provides rapid, life-saving metabolic energy, while the plant polymer passes through the human gut entirely untouched as dietary fiber. We routinely group these fuel sources together as carbohydrates, placing them at the foundational base of our nutritional models. But those broad dietary categories obscure a highly rigorous set of biochemical rules operating at the molecular level. The physical distinction between an impenetrable plant wall and a deployable energy tank is determined entirely by the laws of stereochemistry and glycosidic linkage geometry. To master carbohydrate biochemistry, we have to evaluate these molecules from the bottom up, starting with the exact placement of a single carbonyl carbon. Biological utility is an illusion. The true functional identity of a carbohydrate is strictly governed by its three-dimensional stereochemical architecture. At their structural baseline, simple carbohydrates are defined as either polyhydroxylated aldehydes or polyhydroxylated ketones. We observe this divide by comparing their simplest forms, glyceraldehyde, an aldose, and dihydroxyacetone, a ketose. In an aldose, the most oxidized carbon sits at the very end of the chain, forming the aldehyde group, designated as carbon 1. In a ketose, the most oxidized carbon is located one position inward, at carbon 2, forming the ketone group. Biochemists classify these sugars using the D and L nomenclature system, which is dictated by the highest numbered chiral carbon in the molecule. In a standard Fisher projection, if the hydroxy group on that specific chiral carbon points to the right, it is classified as a D sugar. When two sugars share the same chemical formula but differ in their configuration at exactly one chiral center, they are known as epimers. Inverting just a single hydroxyl group, like the difference at carbon 4 between glucose and galactose, creates a distinct molecule with entirely different physical and chemical properties. At the monomeric level, the spatial swap of a single hydroxyl group establishes an entirely new biochemical identity. This structural divide directly dictates clinical outcomes in medical engineering. In chronic wounds, a destructive protease enzyme called elastase accumulates. It breaks down the extracellular matrix and prevents the tissue from healing. Researchers hypothesized they could clear the wound with a carbohydrate-conjugated cellulose glucose, acting as a molecular sponge to selectively bind and sequester the elastase. They tested two specific variations, bandages coated with aldohexose sugars and bandages coated with ketohexose sugars. This graph plots the remaining elastase activity in the wound environment on the y-axis against the mass of treated fiber applied on the x-axis. The aldohexose group, galactose, allose, and mannose, produces a relatively flat trend line. They leave a high concentration of active, destructive elastase in the wound. Conversely, the ketohexose group, including fructose, sorbose, and tagatose, shows a sharply descending drop in activity. That lower remaining activity means the ketohexose sugars successfully bound the elastase, pulling it out of the solution and neutralizing the threat. The chemical makeup of the two bandage groups is nearly identical. The only difference is the placement of the carbonyl carbon. A simple chemical shift of a carbonyl group from position 1 to position 2 drastically optimizes a sugar's capacity to engage in carbohydrate-protein interactions. Within the aqueous environment of a cell, these straight-chain sugars rarely maintain their linear form. They are inherently unstable. A monosaccharide possesses a nucleophile in its hydroxyl group and an electrophile in its carbonyl group. This sets the stage for a spontaneous intramolecular reaction. The hydroxyl group on carbon 5 attacks the carbonyl carbon at position 1. This attack forces the molecule to snap closed, forming a cyclic hemiacetal. The former carbonyl carbon is transformed into a new chiral center, named the anomeric carbon. Because the nucleophilic attack can occur from two different trajectories, it creates two possible epimers, known as anomers. If the hydroxyl group on the anomeric carbon points down in the axial position, it forms the alpha anomer. If that hydroxyl group points up in the equatorial position, cis to the carbon 6 group, it becomes the beta anomer. Exposing these rings to water causes them to spontaneously open and close, constantly shifting between alpha and beta forms in a process known as mutarotation. This cyclization stabilizes the sugar, while simultaneously providing the specific architectural docking port, the anomeric carbon, required to build complex biological matrices. Constructing a complex carbohydrate requires a dehydration reaction between the hemiacetyl ring of one monosaccharide and an alcohol group on a second sugar. This forms an acetyl connection, known as a glycosidic bond. The exact angle of this glycosidic bond dictates the ultimate fate of the polymer. When glucose connects using a Ba1,4 linkage, alternating monomers flips, allowing tight hydrogen bonds to lock the chains into a rigid structural matrix, cellulose. Humans lack the specific cellulase enzyme required to hydrolyze this exact beta angle. Consequently, cellulose passes through our digestive system untouched. Starches, like amylose, utilize an alpha-1,4 linkage. Human digestive enzymes can easily cleave this distinct angle to extract the glucose monomers for energy. The difference between an indigestible dietary fiber and a rapidly deployable energy source is simply the fractional rotation of a single chemical bond. These stereochemical rules culminate in the primary energy storage molecule of animals, glycogen. The core backbone of glycogen is built utilizing those easily digestible alpha-1,4 glycosidic linkages. Glycogen features a major evolutionary upgrade, the frequent addition of branching off the main chain. These branches rely on an alpha-1,6 linkage, occurring approximately once for every 10 glucose molecules. This hyperbranching optimizes the physical properties of the molecule, drastically increasing its solubility within the fluid of the cellular environment. Metabolically, branching creates a massive number of terminal, non-reducing ends along the perimeter of the polymer. This architecture provides multiple simultaneous docking sites. During sudden metabolic demand, glycogen phosphorylase attacks these ends, cleaving off dozens of glucose molecules at once. From the precise nucleophilic attack that forms a single hemiacetal, to the elastase binding capacity of a ketohexose, to the massive branched architecture of glycogen, biological utility is nothing more than an uninterrupted chain of stereochemical logic.